All right, well, I think we can go ahead and get started, and anyone who's joining later can filter in at that point. So, good evening, everybody. My name is Sarah Neckerman. I am the program manager for adult programming and events here at the Arnold Arboretum, and I'm facilitating today's discussion. This program is glass dried and digitized, preserving plants for science and art. We have today with us three wonderful panelists who are going to take a deep dive into these different ways of preserving art for research, for study, for artistic appreciation. So we have Jenny Brown from the Glass Flowers at Harvard University. We have Jenifer C. Lopes, who is a researcher at Harvard University Herbaria. And we have Sonia Ralston, who is with Northeastern University. So I'm going to tell you a little bit more about each of them, and then they will each have some time to give you some more uh, information about the work that they do and how they each kind of contribute to these different ways of preserving plants. And then we'll have some guided discussion, and then at the end, we'll open it up to Q&A. So feel free to put your questions in the chat as we go along, but we'll be addressing them kind of at the end of the talk. So to give you all a little bit more information about our panelists, so we'll start with Jenny Brown. Jenny Brown became the collection manager for the glass flowers at Harvard University in 2012. She's worked on many exciting projects since then, including co-authoring Glass Flowers, Marvels of Art and Science at Harvard, completing the most extensive exhibit renovation in the collection's history, and co-curating special exhibitions within the Glass Flowers Gallery. Jenny's background in art and library science, combined with her experience assisting renowned glass artist Tutsinski, prepared her to work with this unique collection. Next, we have Jenifer C. Lopes, who is a researcher with the Harvard University Herbaria, and she's using herbaria specimens to study the plant family Valoisiaceae. I think I'm not pronouncing that correctly, but Jenifer will pronounce it more correctly when she comes on. Jenifer has worked with herbaria since 2007, studying at the University of Sao Paulo Herbarium during her undergraduate, master's, and PhD programs. She has spent most of her career since then associated with that University Herbaria and other herbaria around the world, including herbaria in Europe, Leiden, Paris, Belgium, England, and the United States. Sonia Ralston is a designer and researcher and is currently a research fellow at Northeastern University, where she does work on botanical modeling, as well as bioindicators and environmental information systems. Sonia creates these really highly detailed digital plant models and is currently working to create a digital herbarium of bioindicator species that respond to soil and air pollution in the Northeast. She's also working on an exhibition at the Arboretum called Ghost Trees, which uses augmented reality to represent digital models of trees that used to be highly valued and beloved at the Arboretum, but which are no longer standing. So that will go up this fall. So I'd like to turn it over to Jenny Brown to begin with, to give us some more information about the work that she does.
Thank you so much, Sarah. Thank you, everyone, for being here. Let me just get my screen going. Sorry, I'm just trying to switch my my view here.
Jenny, you might try display settings.
Yeah, I can't. I feel like the little bar at the top is blocking that. Hold on, let me try that again. I'm so sorry, everyone.
Technical problems are a part of life. We'll never avoid them. There we go.
Hey, does that look right? Can everybody see my first slide? Yes?
That looks great.
Okay, great. Thank you so much for inviting me to participate in this panel discussion. I'm going to tell you a bit about the history of the glass flowers at Harvard University, and I'm going to give a brief overview of a major exhibit renovation that occurred in 2016. So the official name of the glass flowers is the Ware collection of Blaschka glass models of plants. And this unique collection has educated students and the public, delighted museum visitors, and inspired artistic and scholarly works for more than a century. The collection includes astonishingly realistic life-size models that look like they were pulled from the ground or cut from a branch. And there are also magnified detail models that show plant parts and anatomical sections. A museum was established at Harvard University to exhibit materials from the zoology, botany, mineralogy, and geology departments. An addition for the botany department was completed in 1890, and it included space for public exhibits in the Botanical Museum. The glass flowers commission began while this addition was under construction. The collection has always been on display in the same place, but there have been some administrative changes. The Botanical Museum collections, including the glass flowers, were eventually transferred to the Harvard University Herbaria, and the Harvard Museum of Natural History was established in 1998 as the public face of the Herbaria, the Museum of Comparative Zoology, and the Mineralogical and Geological Museum. So today, the glass flowers are on permanent exhibition at the Harvard Museum of Natural History, and they belong to the Harvard University Herbaria. Professor George Lincoln Goodale commissioned the glass flowers as a teaching collection and a museum exhibit. He was a botanist and the founding director of the Botanical Museum. Goodale was inspired by the displays of mounted animals in the Museum of Comparative Zoology, but creating a similar exhibit about botany was difficult because plants don't look lifelike when they are preserved. Most scientific models were made from paper mache or wax, but Goodale wanted more exceptional models than ones made from those materials. Here you can see how effective the glass models are. Goodale saw models of marine invertebrates made from glass on display in the Museum of Comparative Zoology while he was planning exhibits for the Botanical Museum. And these glass models were made by Leopold Blaschka and his son Rudolf. Like plants, these soft-bodied organisms don't look lifelike when they are preserved, but the Blaschka's glass models are realistic representations that can be studied in classrooms and displayed in museums. Goodale thought the Blaschkas could produce equally realistic plant models for the Botanical Museum. The Blaschka's glassworking lineage is believed to trace back to 15th century Venice, and their craft was passed down through multiple generations. Leopold learned glassworking from his father, and he also studied painting, jewelry making, and metalworking. Rudolf started working with his father in 1870 when he was 13 years old, and he officially joined the business in 1876. The Blaschkas lived in Bohemia, which is in the present-day Czech Republic, and they moved to Dresden, Germany in 1863. Leopold had the idea to imitate marine invertebrates in glass when he saw jellyfish while traveling to the United States in 1853. He made his first marine invertebrate models 10 years later after moving to Dresden. Museums and private collectors started ordering models, and over the next 2 decades, the Blaschkas established a successful business producing invertebrate animal models for institutions all over the world. Goodale visited the Blaschkas in 1886 to ask if they would make glass models of plants for the Botanical Museum. They were reluctant at first, but eventually agreed to the commission. The first shipment was damaged during a customs inspection in New York, but the broken models were promising and they were shown to potential supporters, including Mary Lee Ware, who was one of Goodale's former students. She and her mother, Elizabeth C. Ware, provided funding to order more models. So with support from the Wares, the Blaschkas divided their time between making invertebrate animal models for other institutions and plant models for Harvard. After 3 years, the Blaschkas wanted to focus on either zoological models or botanical models, and they signed a 10-year contract with Harvard to work exclusively on the glass flowers in 1890. Leopold died in 1895, and the contract was updated for a one-person operation. Many other exclusive contracts with Harvard followed, and work on the collection continued, always with the Ware's support. An agreement to make some models in 1886 turned into a 50-year-long project. The final shipment of models was delivered in 1936, but Rudolph was still working on the collection when he died in 1939. Over 50 years, from 1886 to 1936, the Blaschkas produced 4,300 models that represent 780 plant species. They only made glass models of plants for Harvard, but their invertebrate animal models are in collections around the world. The glass flowers were made by Leopold and Rudolf Blaschka alone. They didn't have assistants or apprentices, and their productivity was so great that about 70% of the collection was completed when Leopold died in 1895. The Wares were devoted benefactors. Mary supported the project until her death in 1937. They also covered all related expenses, including the exhibit cases that are still in use today. The collection is a gift in memory of Elizabeth's husband and Mary's father, Dr. Charles Eliot Ware, who graduated from Harvard in 1834. The Blaschkas were exceptionally talented and innovative. They used lampworking to make the models. This is a glassworking technique in which glass is melted in a flame and shaped when it is soft. Lampworking is also called flameworking or torchworking because gas-fueled torches are used instead of oil or alcohol lamps today. The models are glass, but wire was used for internal support. Tubes of glass were strung on a wire armature to form the stem, and leaves, petals, and other parts were attached with animal glue, or pieces were fused together in the flame. Early models were made from clear glass and painted after they cooled. Rudolph experimented with materials and methods after his father died. His greatest innovation was making glass enamels and using them to color models instead of paint. Gelatin was used to create leaf veins and other details, and organic materials like animal glue and natural resin varnishes were applied to the surfaces of the models to reduce shine and mimic different textures. All of these organic materials were applied after the glass was shaped and cooled. The glass flowers are realistic and scientifically accurate because the Blaschkas studied living plants. Other reference materials include their own drawings, botany textbooks and publications, and preserved plant specimens. The Blaschkas studied zoology and botany, and they applied this scientific knowledge to their work. Most scientific models in the 1800s and early 1900s were made of paper mache, and others were made of wax. The Blaschkas' use of glass for scientific models was innovative and unusual. Glass wasn't, and still isn't, a common material for scientific models. I'm aware of only 2 other collections of glass scientific models. The Whipple Museum at the University of Cambridge in the United Kingdom has about 100 glass models of fungi that were made by Dr. William Dillon Weston from 1936 until his death in 1953. And the American Museum of Natural History in New York City has a collection of glass models made by Herman O. Mueller. He made about 1,000 glass models from 1903 until his retirement in the 1940s. Dillon Weston's and Mueller's models are beautiful and educational, but their output doesn't compare to the thousands of models produced by the Blaschkas. Of course, scientific models started being made from plastics in the 20th century. The glass flowers reflect scientific knowledge in the late 19th century and how it was taught. There was an emphasis on studying the form and structure of organisms to understand their evolutionary relationships. The glass flowers illustrate plant diversity, and the exhibit is arranged so comparisons can be made among different plant families. Each species is represented by a set of life-size and detail models. The detail models include magnified cross-sections of ovaries and enlarged plant parts. These detail models are especially useful as teaching tools because they show plant forms that often aren't visible without dissection and magnification. Scientific models like the glass flowers became less relevant for teaching as research changed in the 20th century. The focus shifted from morphology to molecular biology and genetics. The glass flowers still educate students and the public about plant diversity, and scholars of botany, art, history of science, and many other disciplines study the collection. The glass flowers also inspire artists like Lila Tabasso and Deborah Moore. The glass flowers have always been a popular attraction. The collection has been on display in the same gallery since the Botanical Museum opened in 1890. Changes and improvements were made to the exhibit after the collection was completed in 1936, but major renovations weren't possible because of the challenges involved with moving and storing the glass flowers. The most extensive renovation of the glass flowers exhibit occurred in 2016. Work started in the gallery after a year and a half of planning. During this time, a new storage facility and a conservation lab were set up, and the collection's first conservator was hired. One of the project goals was to maintain the exhibit's character and historic charm, while necessary updates were made. The exhibit was completely emptied for the first time in the museum's history. All object handling was done by me and the conservator, with some assistance from local art handlers. Here are some more before and after photos. The biggest change was that the floor plan was redesigned. As you just saw, the layout divided the room into 3 sections, and the new floor plan is more open. Continuing to use the historic cases helped us maintain the exhibit's character. Most cases are over 100 years old, and all of them were restored. 8 new cases were designed for changing exhibitions within the Glass Flowers Gallery. Having space to bring models out of storage for special exhibitions lets us show more of the collection and gives visitors reasons to return. The models were rearranged according to the angiosperm phylogeny group. This is a contemporary classification system that reflects current knowledge about the evolutionary relationships among plants. The previous arrangement was based on the Engler plant classification system, which was widely used in the early 20th century. Taxonomy was updated and all labels were redesigned. And content about plant anatomy, composite flowers, and other botany topics was added. The exhibit still feels like it's in a Victorian museum, but the science is current. The HVAC system was replaced and upgraded. Climate control is important because the glass flowers are susceptible to temperature and humidity fluctuations. The new HVAC system keeps the temperature and humidity within a safe range to slow deterioration and preserve the collection. A new LED lighting system was also installed. Some pigments that were used to paint early models are light sensitive, and the colors are susceptible to fading and shifting. The light levels in the exhibit are controlled to minimize light damage. Other improvements were made, which include painting the room with bright neutral colors and uncovering and refinishing the original wood floor. The exhibit was closed for only 6.5 months. About half of the collection is now on display. The exhibit renovation was the catalyst to start a formal conservation program. The glass flowers never had their own conservator. Research and conservation treatments were done on an as-needed basis by conservators from the Harvard Art Museums and the Peabody Museum of Archaeology and Ethnology at Harvard. Scott Fulton became the head conservator for the glass flowers in 2015, before the exhibit closed for renovation. Scott was a senior objects conservator at the Peabody Museum for many years, and he started working on glass flowers projects in 1997. Conservation was an important part of the exhibit renovation because many models needed to be stabilized, cleaned, repaired, and/or remounted before going back on display. There wasn't enough time to perform conservation treatments on every model while the exhibit was closed for renovation, and the conservation program is ongoing. This has been an exciting and productive time for the collection, and most importantly, significant progress has been made in the care and maintenance of the glass flowers so they are preserved for future generations. Thank you so much.
All right, thank you so much, Jenny. Next up, we have Jenifer C. Lopes, who's going to tell us about her research at the Harvard University Herbaria.
Are you seeing my screen?
Yes, we see the screen.
It's a great pleasure to be here with you talking about what is a herbarium and the use we can make of the scientific collection. And yeah, let's start. An herbarium or herbaria in the plural is a collection of dried, preserved plant specimens and associated data used for scientific purpose. Then the herbarium is a natural history collection, but it's mainly for scientific research. Then we don't have, it's a museum that is not open for the public, for the general public, but it's open for other researchers. And usually it's very common that a researcher goes to other herbarium in different countries sometimes to study the collection in that place. And what is a herbarium specimen look like? Herbarium specimen is a dried and pressed plant mounted on paper. And it's important nowadays that this paper have an archival quality, meaning that they are acid-free and that they can preserve less long. Our herbarium specimen can last like hundreds of years. Some of them have that are still very important scientifically, have up to more than 200 years. They were collected in the 19th century, for example. One thing that is important to know is when we describe a new species of plant, we have to have like a specimen that is deposited in our herbarium to be associated with this name. Then we can check the name and the specimen to see how this plant look like. And this specimen that is associated with a scientific name is called the nomenclatural type. And these specimens are very important. Then for a species that were described like a long time ago, for example, in the 19th century, they still have this nomenclatural type somewhere in our herbarium to open to the other researchers to go there and check. And the herbarium also include associated collections. Here is a picture of the herbarium in Paris. I like this picture because it's very beautiful with many colors. The colors, the cabinet it's yellow, and we have like folders in different colors. The herbarium specimen is flat, and then we can put many specimens one on top of the other, and in folders, and store them in these cabinets. But for other material that we cannot store in this way, we have these associate collections, like the wood collection, the fruit collection. For example, there are fruits that are too big to be pressed and stored in this way. Then we can store them in boxes. And also we have the silica storage material. Nowadays, people are aware that it is important to preserve leaf tissues for extracting DNA and do other kinds of analysis. Then besides collecting the specimens that will go to the herbarium, they also collect the leaves and store in silica gel for make it easier to extract DNA. And we also have the fluid preserved flowers or fruit. They are usually preserved in ethanol. And this is a way to keep the tridimensional shape of the flowers, because when we press a flower or a fruit, sometimes we lose some of this three-dimensionality, and it is easier to preserve than in fluid, And we also have the microscope slides for people interested in analyzing the pollen grains of the flowers. And each herbarium has its own acronym, and these acronyms are can be searched in the Index Herbariorum. That is a system developed by the New York Botanical Garden. For example, here I'm presenting the 15 largest herbaria in the world, and each one has its own code. And for example, the largest herbarium is the herbarium from the Royal Botanical Gardens in the United Kingdom, and its acronym is K. And we have like the herbarium in Paris and the New York Botanical Gardens also, with almost the same number of specimens, around 8 million specimens. And the Harvard University Herbaria is also among these 15 largest herbaria in the world, with around 5 million specimens. And one thing is interesting is that the Harvard University Herbaria is the largest university herbaria. All these other largest herbaria, they are herbaria from research institutes or botanic gardens, such as the New York Botanical Garden or the Missouri Botanical Garden. And we call herbaria because the collection that is now in a single building, it used to be spread all over Cambridge and Boston. For example, the Arnold Arboretum had his own herbarium that the acronym was A, and there were also an herbarium specifically for orchids, and another one, the Ames herbaria, and the other one, the Gray herbarium, that was mostly collections from South America, and the economic herbarium of Oak Ames, and they were in a couple of years ago, they were all placed together in a single building. And the glass flowers are also part of the Harvard University Herbarium, as Jenny just mentioned before. And now I'm going to talk about how we collect and preserve the herbarium specimens. First, there are different ways to collect plants depending on the habit of this plant. You can use a pruning shears, or sometimes if you are collecting a very tall plant, we may need to ask someone to climb this tree to collect a piece of the tree. Here I'm collecting a tree with a telescope tree pruner. Or, for example, I learned recently how to collect bromelias. In fact, you have to, the most effective way is this way. This person is showing with with his foot. And it's very important to annotate characteristics of the plants and also its location, and take as much as many records you could have during this process. For example, during the process of drying the plant, we lose a lot of information, and some of them are, for example, the color of the plant and the locality of the plant. Then we have it's always important to take photographs if the person can, and also take the geographic coordinates and the exact place where this plant is collected, and also take notes about the plant itself. For example, if it's a tree or if it's a herb, or the color of the flowers, and if it's fragrant. And all this information that is associated with the plant is very important nowadays, because the herbaria are being digitized, and this information about locality, for example, are being very important to people working on geographic distribution, modeling climate change, and I will talk more about that towards the end of the presentation. And here is a specimen and its label. And a label should have the name of the species, like the binomial, genus, and a specific epithet. Then a plant description, such as I mentioned, if the flower is fragrant, the color of the flower, and other important characteristics that may lose during the drying and pressing process. And also the location is very important, because for knowing the exact place where a plant occurs is also part of the scientific knowledge, because usually the plants are constrained to certain ecological and geographical range. And then this precise annotation of the place where the plants occur is also important for multiple analysis. And it's also important to have the date of the collection and also the collector, because these are ways that we can trace back the specific specimen when we are citing this material for scientific purpose. And now I'm describing how we preserve that and make the herbarium specimen I show in the beginning of the presentation. Then here are some students, and here my supervisor, my PhD advisor, collecting plants in the field. The best way is to press the plants when they are still fresh, when we just collect it. And the way that we do that is we do that for hundreds of years, because it's very efficient and simple. And as we do that, we take up a piece of the plant that is that fit in the mounting paper that we will use to preserve the specimen. And usually we use newspaper and fold it and put a cardboard between each plant with newspaper and make this press. And it's sometimes we have to press it very well, and we can like do as my student is showing, to make sure that the plant is pressed adequately, because this is very important in the second step of the process, that is drying the specimen. And for drying the specimens, we usually include the presses in the oven for several days. Sometimes it's varied the temperature, but sometimes it's from 3 to 5 days. And there are, of course, there are certain species that they have more water, and we will have to dry them longer. And to end my talk, I want to mention some of the uses of the herbarium specimens. This was part of the work of the professor Charles Davis, that is from the Harvard University Herbarium, and here he produced this nice figure showing the proportion of uses herbarium specimens can have for scientific purpose. The most common are the taxonomy uses. For example, if we want to know how many plant species occur in the United States, we would have to have records of plant species, preserved herbarium specimens that were collected in the United States for being able to prepare this list. Then every time that we need to know the number of species in a certain region, we have to rely in the records from herbarium specimens. Another thing that is very important is if we wanted to know, for example, the distribution range of a particular species. If we want to know the distribution range of, for example, for the red oak, we will need to look in the herbarium where these plants were collected. And every record will help us to understand the distribution pattern of these species. And another thing that is very important is the description of new species. My colleague and I, we were working on the flora of Papua New Guinea, that is an island close to Australia. And this area is still under studied. And we were looking at herbarium specimens that were collected in the beginning of the last century, and the 1970s and 1940s. And when we look at these specimens, we could discover at least 7 new species of plants. In other words, plants that they don't have a scientific name. And if a plant doesn't have a scientific name, it means that they don't exist for science, and we don't have a way to protect them or to know more about them. Then their herbaria, they are important also for discovering biodiversity. But beyond that, nowadays we can use the historical records of herbarium, because plants have been collected for hundreds of years, as I mentioned, to know how climate change affected the distribution of plants. And another way is also to use these dry preserved leaves to extract the DNA and try to understand the relationship, the evolutionary relationship between plants. This year was published a study investigating the evolutionary relationship of all flowering plants, and most of their data rely on DNA extracted from plants, from dried plants in herbarium specimens. And for the future, we still don't know which the possible utilities their herbaria can have in the future. Then it's important to preserve this collection for allowing us to understand more about biodiversity.
All right, thank you so much. Next up, we have Sonia Ralston, who's going to talk about her art and research with botanical digital modeling.
Requisitely asking if you can see it. Okay, great. Hi, so thank you so much for the invitation to be here. It's wonderful to be able to share my work alongside Jenifer, and thank you so much to the Arnold Arboretum for having me do this collaboration that I'll talk about a little bit later. Let me just optimize, there we go. So I thought it might be best to talk a little bit about how I got here and who I am. So I grew up in the West Coast near Vancouver in Canada, and this image is of a forest near where I grew up. It's a plantation forest built after significant labor and logging protests on the indigenous lands of the Katsi, Semiahmoo, and Kwantlen First Nations. I work as in architecture and landscape, and my landscape design, or these things are together. But I've always dealt with how the environment is kind of memorialized through different media, and have been especially interested in forms of environmental data and plant record keeping. So my work investigates how plants and information systems collide and the ramifications this has socially, politically, and spatially. So in this talk, I'll just talk about a couple of projects that I've used these digital plant models in and why that was important. And then in the last part, I'll talk about this collaboration I've done with the Arnold. So my work here with these digital plant models started as a graduate student when I was at the Harvard Graduate School of Design. And I was encountering a lot of different ways that ecology and technology were intersecting. So my thesis, which there are many images, but this is one of the many videos, was titled Uncommon Knowledge, Practices and Protocols for Environmental Information. And the idea was that it was situated on the Columbia River at the edges of Google's first hyperscale data center. And the idea here was that there's a large amount of information infrastructure that's required to power something like Google. Think of like AI technologies or things that power algorithms on Instagram. But that has a really material effect. So I was interested in the way that water extraction that's used to power kind of information systems actually has an effect on local ecology. And so this project, which was a speculative kind of project, which is something that artists and landscape designers do as a way to kind of research what it means to represent the future, what that kind of has to do with this different forms of extraction. So as part of this project, I wanted to think about plants and the landscape itself as this kind of form of information infrastructure to indicate environmental change. So to me, and probably to many of you who are listening, the planet is its own kind of robust form of naturally intelligent knowledge, but many of us, maybe at least those of us who aren't horticulturalists or attuned to the intricacies of plant life, don't really have the tools to understand them. So what I was hoping to achieve by this was to have multiple kind of plants that were modeled in these digital models that had different purposes in the sites. So some of them were hyperaccumulators that were supposed to suggest the presence of pollution. So they might know that as a bioindicator or a sentinel species, but also kind of performing social functions. So how different plants and their connections to different indigenous traditions would be kind of important sources of social knowledge. So integral to this kind of proposal and this thesis work was to really think about how plants are like a literal and material form of information. And I chose to represent this through kind of handmade botanical modeling, so not algorithmic methods. So I'm literally drawing all of these different little parts of the plant in the computer. So many of these plants are also endemic to this region and didn't necessarily have kind of digital models of them, because you might recognize a lot of digital models of plant from video games or movies or things like this where they have them. So this was my kind of start. So part of my decision to model these plants in detail was also a response to the kind of literal and conceptual flattenings that happens in large scale environmental data systems like in herbaria. So plants here are literally data points, and then they're sheets of paper, but they're organized under kind of Western ontologies of information and are held in kind of Western geographic centers. So part of this form of work was to try to think about how can we think about plants themselves as a form of information, as opposed to something that's been categorized into these kind of colonial systems. So each of the representations of the plants in this project was really aimed to thinking about, like, again, plants as information and not just kind of as a backdrop or an ornament in the landscape, and really try to encourage viewers to think about the environment as kind of something to engage with and to learn from. So that's why you can see these kind of type sheet drawings where the plants are drawn almost like in this technical kind of infrastructure thing. So they're performing kind of an infrastructural thing. So I modeled all these plants by hand again in the computer, but not just as a way to understand their structure and patterns, but in a way to try to really get close to them in this kind of digital medium. So the reason that I'm distinguishing the way that I make plants from the algorithmic methods, which you see in this image here, is because that's actually a really robust area of scientific research. So there are lots of softwares, like I mentioned, that are used for, making animation trees and plants and things for animation that are being built off the research by algorithmic botanists like Aristid Lindermeyer, who built the mathematical models to create kind of these digital plant models. So this is the work of science, and my work, by contrast, is different because I don't use algorithms to simplify the process. So to this end, this kind of all this work initially led me to the question that as a designer is really important to me, to think about how plant life as it appears in the form of kind of like proposals for parks, like as you would see in like a rendered image, it's really just to think about kind of the historical, social, and cultural relationships that are embedded within those digital plant assets or models. So how can we focus on plants as a kind of central form of experience and inquiry that can lead us to learn more about the world? So that's kind of my main premise in this work. So I'll speak quickly about a recent project that I did that used these digital plant models, although in a bit of a different way, to think about plants as a kind of form of data again, but also to reflect on the idea of memory and aftermath. So as part of Data Through Design, which is an exhibition that takes place in New York City, where artists and designers are asked to use a data set from New York City's open data and create an artistic representation of it. So I used a species entitation study, which is essentially a kind of plant survey, but designed for urban scales, to develop digital models of the plants to show the different changes to the plant communities that were happening over time, in this case in Pelham Bay Park. So the plant models were used as material to develop visualizations, which you'll see a little bit in a second, but also to think about these plants again as informational entities that evidence landscape change and also kind of social engagement with it. So in 2017, a large white oak, which was believed to be over 200 years old in Pelham Bay Park, fell down as a result of a big nor'easter storm. So you can see it's beautiful, gigantic branches. It's difficult to understand the scale of it until you're there. And then there it is, fallen over. But as I said, it's difficult to understand the scale, but what was most shocking to me actually is that I visited the site of the tree that had fallen this past spring, and it's completely unrecognizable where it is now. So the clearing where the oak once stood, the trees, the shrubs, and the herbaceous plants have kind of responded to the hole left in the canopy. And all of this brush that you see on the bottom is Multiflora rosa, which is an invasive species, and it's completely taken over the site, making native species kind of few and far between. So this is something that, happens all across the Northeast. This is, again, like a particularly bad problem that's faced in this part of New York, which is up near the Bronx, which has faced a lot of disinvestment over many years. So the way that I ended up making these digital plant models part of this story was that I used the entitation maps, which you can see that's made from an aerial survey. This is an image from 1986 on the left, which essentially what they did to create this entitation map was that there was a photograph taken from a plane. Someone delineated kind of on maps roughly where they thought there might be different plant communities and kind of made little circles of them on the map. And then people went out on the ground and cataloged all of the plants within those units and ground truthed, or just made sure that those were actually indeed separate units. And so it required a huge amount of labor and record keeping. And in this particular unit where you can find the granny oak that I showed you earlier, which is unit 1001, there are 18 different plants. So this isn't quite 18, but this is, I developed a digital catalog of plants from this survey in an effort to really depict how the landscape had changed and how the plants themselves are evidence of those changes. So here, you can have anything from a chive, which is in the top left, to porcelain berry down in the bottom right. So it's kind of a mix of different species here. So it's not necessarily just species that would be considered to be beautiful ornamental plants or native plants, but also things even like porcelain berry or garlic mustard that are invasive plants that are also damaging the landscape. So it's really trying to capture this moment. And what this ended up culminating into was 2 different videos, as you saw in the first image that I showed of this project, was to show the kind of original site based on the survey. So me trying to use the data to reconstruct this place. And I had it kind of inside this point cloud in order to give it kind of like a bit of a sense of the space. And so this is my imagination of what it might look like. In this video depicting the aftermath, I wanted to capture this felled oak. As you can see in like the front, there's a bit denser point cloud where I tried to emphasize, the scale to which the multiflora rose took over the site and the tree. So the idea here was that, the big fallen over tree becomes part of this like LiDAR data set or like point cloud that you can understand topography with. But it's really taken over and changed a lot just by the loss of one tree. So I really wanted viewers to think about deeply as plants as a kind of data point that's indicating things about their local ecology. And it's not just embedded in systems of, plant life, but it's also, embedded in the memories of people who labor to record and maintain it. So plants, as I'm sure this audience believes, or at least knows, are part of a much larger and rich social and cultural history than a line in a survey or a page of a survey can really adequately convey. So here, I'll just finish up by previewing a project that will soon be available to view as a walking exhibition in the Arnold Arboretum titled Ghost Tree, which explores deaccession trees or dead trees that are part of the Arboretum grounds. So before I start this, I just wanted to thank Sarah Neckerman for her work collaborating with me on this. This emerged through conversations, and the plants that were selected were all selected by Sarah and the team. So thank you so much. Okay, so this is just the idea of this project is to think about deaccessioned or dead trees on the Arboretum landscape and try to give people an opportunity to visit them, or at least like a digital version of them, as they walk through the Arboretum. So this might have been something that some of you have seen at some point in your time at the Arboretum, but this is over on Conifer Walk, which is my favorite part of the Arboretum. And this past April, nothing was sadder than seeing my favorite grove of trees succumb to wind damage in a storm. So trees in the Arboretum, they're part of a collection. They are living trees, but that also means that they die or they need to be removed for various reasons like disease, or they fall in a storm and this kind of thing. So again, these are trees that were important or socially, culturally important in the Arboretum, and we tried to recreate them. So without further ado, this is the corky tree, or it's a cork tree. And it was really famous because its limbs were climbable, and large groups of school children would come and pose for photos on them and sit on the limbs. It was the only tree in the Arboretum that you were allowed to climb. And in 1995, during one such photo, the limb collapsed and the corky was removed. So here's a kind of digital reconstruction of it. And what was really interesting while working through this model was that there are lots and lots of photos, because there are so many photos of school kids on the tree. So I had a lot of different angles to be able to try to think about the structure of the tree and how to try to make it kind of work as best I could to adequately kind of reflect that moment or a few moments in time where there were photographs available of the tree. Another interesting part of modeling the cork was to really think about how to get that kind of quirky, coarse bark. So that was an interesting challenge. The second tree, it's a tree that still exists today. It's a Donald Wyman crabapple. And I wrote that it's deaccessioned in 2018, but it's not really. It actually still exists as a stump, which you can see on your right. It's not really a stump. It's still a little bit alive. And this is just so yes, it's been damaged by a couple of storms. So in this way, you can revisit its version before 2008. And what was really interesting about working on this tree was to try to really make it appear full of fruits without getting too much kind of repetition. So often I'll have like a couple of models of different branches, and then I'll just kind of disperse them. only a couple more trees and then I'll be over. But so here, this was a winterberry holly. It was removed from the main next to the main gate of the Arboretum. It was a really difficult decision, but there was a signature red Dawn redwood tree behind it, which you probably have recognized if you've walked up to the Arboretum before. And here we let the winterberry holly take over and block it again in this slide. So yes, what was really interesting again here was to try to think about the different structure of the tree and how to get that kind of density of branches in order to reflect it. It's a little bit harder to make it as accurate because there's only one image. You are probably familiar with a kind of American elms and Dutch elm disease, which has damaged and kills many, many elm trees in this area. But this particular elm tree predated the Arboretum's founding, and you can see it in hand-drawn maps from that area. It's not exactly clear when it came down, but it was deaccession in the early 20th century. And here I was really interested in trying to get that kind of twisting motion of elm trees and to really come through. And this is the final tree that will be part of ghost trees, but it came down in 2018 due to beech bark disease, so another kind of disease. And it was, I think, part of the notes of this and what was important here for the Arboretum was to show this as a kind of the potential destruction that introduced pests and diseases can bring to forests around us. So again, it's really, I think this is a really helpful example to think about species as sentinels to understand environmental change. And so in short, all these trees are a really good way to connect us to memories of plants gone by, whether you had clambered up the Corky as a child, or you've heard of the loss of elm trees, or you've witnessed the removal of one of your favorite trees in the Arboretum, at least in spirit or digitally, you can revisit some of these trees as part of an exhibition here. So if we try to think about plants as a kind of information system or repositories in and of themselves of important cultural memories as well as kind of scientific knowledge, we might be able to better connect to our own memories and observations of environmental change. So thank you so much.
Amazing. Thank you so much. Thank you to all three of you for those wonderful slides. So now we all have kind of a high-level view of each of these forms of preserving plants, right? We have the glass flowers, we have the dried and pressed plants in herbaria, and we have these 3D digital models. So they all use different materials, they have different histories, there are different processes for creating them, but they all exist to preserve plants in some way. So now I want to dive a little bit deeper into the similarities and the differences between these forms of plant preservation. So I have a number of questions for our three panelists to start uncovering kind of the benefits and the drawbacks of these different forms and how these different preservation methods intersect. So the first question is, what is the benefit of preserving plants in these ways now that we have such high quality photographs? And any of our panelists can answer these as they see fit.
I will reply to it. This is something interesting. Nowadays, there is a big process in the herbaria digitization, mainly for photographs and also the transcribing the labels that are also important. But besides that, there are many things that we cannot see in a photograph, such as the details of the plant, and also we can, as I mentioned during my talk, use parts of the plant that we call destructively sampling to, for example, extract DNA and investigate the genetic diversity in the plant, and also using it for understand the evolutionary pattern among species. My opinion is that the photos of these specimens, at least for herbarium specimens, they are good for showing people that these specimens exist, and for them to be aware of the a variety of species and specimens that we have in the herbarium. Because before that, the only way to know what actually exists in a herbarium were people going there and going through the cabinets. Then I think that the photos, they are a way to show the specimens that we have and the possibilities of use that we have, instead of not actually preserving the specimens in the way they are for preserved for centuries in different herbaria.
Wonderful, thank you.
I just want to jump in because the glass flowers were made, while photography was still an early and emerging technology, and photography wasn't widely accessible to everyone. So the glass flowers, they gave people an opportunity to see uncommon plants, on exhibit in the then Botanical Museum. So, the glass flowers were, there wasn't really an option to have photographs of all of these plants. So going to see the glass flowers at the museum was a chance for people to see all of these plants. And today it's an opportunity to see, all of these plants in bloom year round, forever.
Absolutely, thank you. Sonia, were you going to add something?
I could, but also if you want to go to the next question, that's okay too.
No, go right ahead.
All right. Yeah, I think this is, I feel like I'm the person who probably in a way has like the least utility, but this is also part of the problem of being a designer, is that, as I mentioned, there are lots of different kinds of forms of, algorithmic modeling of plants that were used to essentially model kind of plant growth or plant growth or plant decay and in different kinds of tools. Again, that's a field of study called algorithmic botany. But I guess what's important to me about the things that I do is that it's actually kind of hard to find kind of bespoke digital models of plants in order to understand them as kind of like three-dimensional, especially trees, as three-dimensional beings, especially if you have a very specific species or like a very specific tree that you want to preserve in some way. So while it's maybe not necessarily kind of useful in the kind of like for scientific study in the way that going to an herbarium and taking a sample would be, but to me, what's really interesting and important about this is that it's really kind of helpful for like a cultural or social impact for people to be able to, for example, with the trees in the arboretum that I'm modeling, to really go and be able to visit those trees that had kind of social importance to them, or have a kind of reflection on the way that the environment changes from when you had first seen it when you were a school child in 1995 to the stump that's on the ground now.
Absolutely. Your point about practicality kind of leads into our next question, which is that all of these forms of preserving plants are not only practically useful, but also beautiful and artistic. So is there value in the beauty of these pieces and in the art of creating them separate from their practical value?
I can jump in on this one. I think, the glass flowers are scientific models. The Blaschkas really, regarded this work as, a scientific endeavor. So I think, first and foremost, they were, objects for science and also artworks. But it is, it is difficult to, you can't, the artistry of the glass flowers is such an important part of this collection. Like you can't, you can't disregard that, you can't, it is hard to just look at the glass flowers purely, for teaching. And I do think too, with the glass flowers, I think, over time, as, research has changed and technology has advanced, today I think the glass flowers are regarded more as these remarkable artworks, which, they certainly are. You know, how the collection is regarded has sort of changed over time. And you know, they do exist in the worlds of science and art, and you really can't, you know, they're both really dependent on each other, I think.
Absolutely. Thank you.
Yeah, I guess just like one way, I'll speaking to kind of like digital plant models in general, as I was mentioning just a minute ago, and also in the presentation, yeah, botanical kind of like plant modeling in digital spaces is really useful for, again, modeling like plant growth, decay, and how different kinds of effects of like different environmental changes might kind of affect that plant. There's all kinds of different research that happens using these algorithms. But something that's also another really interesting way that digital plant models are being used is for not just kind of rendering the future, which is what a lot of like landscape designers, if you ever walked by a big building with like a big image or a big building site, sorry, where they're about to construct something and they have a big fancy image, like all those trees are digital models for the most part, right? So they're really helpful for imagining kind of like what the future of a place might be like by using different plant species, although don't trust architects on having the correct plant species in the models. But another thing that is really interesting, that's an interesting use of these kind of digital models, is actually digitally reconstructing archaeological sites. So there are a lot of different kinds of, like, again, more like social science uses or design uses for these digital plants. But again, this is the realm of the scientists, as in algorithmic botany, and I keep myself in the realm of designers and artists.
Thank you. All right, so our next question is, each of these forms of preservation help researchers, students, and the public to better understand plants, but they all have their own strengths and their own limitations. Do any of these forms of preserving plants fill a gap or a need left by the others? In other words, why is it important to have all three of these plant preservation methods?
Yeah. I think they have different purpose, yeah, and they are complementary. The way we preserve plants traditionally, like pressing and drying, they have mainly a scientific purpose and an educational purpose too. And the glass flowers, they were, as Jenny mentioned, they were previously for teaching purpose, but now they are also valuable as a art piece. And the digital model, as Sonia mentioned, they have like also a way to connect people with the plants that existed in a place and are not there anymore. Then I think they are all complementary and they have different purpose that make the way we see plants in a more in a different way that fill all our needs as human beings.
Thank you.
I think one of the greatest things about the glass flowers is, you know, yes, they were commissioned as a teaching collection and they're museum exhibits. So it is, you know, the glass flowers exhibit at the Harvard Museum of Natural History is, you know, it's outreach. It's getting the public to, you know, engage with botany and, you know, learn about plants and, you know, if, you know, I'm trying to think of other, you know, really like strictly, you know, botany kind of exhibits in other museums. And I do think that the glass flowers, you know, the public space that they occupy, I think is really important. And, you know, how visitors engage with them. You know, we get visitors from all over the world and, you know, local residents here in Massachusetts, you know, they return, you know, repeatedly to enjoy the collection. So I definitely think that, you know, having this public space for the collection is a real benefit. like as Jenifer said, the Harvard University Herbaria is closed to the public, and it's used by researchers. Things aren't like on display in the herbaria, and depending on I hope, Sonia, I hope that we get to roam around the arboretum and look at your ghost trees. That sounds really amazing. So I think just having these public spaces to engage with the public about botany is really important, and the glass flowers do that.
Definitely. Sonia, was there anything you wanted to add to that? Okay. Awesome. So kind of related, is there anything that you can learn from the work of the other panelists here tonight that you can bring back to your own work and your own collections?
I can start. I mean, it's my favorite is talking to like botanists or people who work in different kinds of botanical collections. It's like my absolute favorite thing to do. I recently was at the Harvard University Herbaria, and I was also like recently at the Arboretum's archives. These are, I mean, I hope that everybody, if you have the opportunity to do this, go and talk to people who work in botanical collections, because there's so much really interesting, not just like even just like knowledge that people have, but there's so much interesting things that people do, and the kind of labor that they do is just such like a different thing than I think for the most part people do on their day-to-day work. So that's one amazing thing to do. And I mean, it's really nice to have the opportunity to listen to both of you, but I would encourage like anyone who's interested in plant collections to go and visit as many as you can, because they're so they're kind of so idiosyncratic and have so many different ways that they work. It's just really, really interesting to kind of ask someone a often very strange question to them, and you get rolling into all kinds of different things. So again, these are really enjoyed the presentations. Yeah. But...
Definitely, thank you.
And I mean, it's kind of cheating because Jenifer and I like actually work together in the herbaria. But just in my work with the collection, I've learned so much from my colleagues in the Harvard University Herbaria. It's been really everybody is smart and kind and generous with their knowledge. And I certainly apply what I've learned about herbaria and about botany, that I've learned through being in the herbaria. I definitely apply that to my work.
Mm-hmm. Awesome. So I have one last question for our panelists. As we're going through this last one, if any of our attendees tonight want to put any questions into the webinar chat, we'll move on to the public Q&A just after this. So our final question, what should the public take away from this talk tonight about the importance of preserving and representing plants?
I can start. I think that the thing that I could get after all the talks is that plants, they are very important for in our daily life. Sometimes we are not aware, but plants are the living beings that allow us to live in this earth. Like they provide us food, and they also provide us the climate for us to live in a safe place, not too hot, not too dry. And sometimes we are not aware of the importance of plants, and seeing different people working on plants in different aspects, we can get to know that the botanical knowledge is still working on, and we can learn from different fields. Like since this is a very interdisciplinary approach that each one of us have, I think. This is what I could mention.
Thank you. Yeah, I guess just as a takeaway, about the importance of preserving them and things like this. I would just encourage, I mean, I'm this is like a little bit of a captive audience in that these are all people, you're all people who care about plants. So I don't need to like wax poetic about how it's really important to just pay attention. But what I think is, I do like an assignment with some of my students who are UI UX designers, so people who design websites who don't usually look outside. And I do an assignment where they have to watch a tree grow for the whole semester. And people get really, really excited and anxious about the growth of the tree and like this kind of thing. And I would say, just in terms of the importance of thinking about or preserving kind of plants, the importance to me is to really build a kind of connection with the environment that you're in and really think about the way that plants are changing. They're living beings. They just operate on a very slow time scale, and preserving those kinds of important cultural memories or spaces that are associated with plants that may not be there forever, just like we won't be here forever, is something that I think is a really important takeaway. Yeah.
Thank you so much. Okay, so I would first like to say thank you for those really interesting, thought-provoking answers. And then I want to open it up to audience Q&A. So we had one question in the chat from Jenny, your presentation on the glass flowers, which was, if 70% of the models were done by 1895, were the rest of the 30% done during the next 41 years? Can you tell us a little more about the timeline of when all of these thousands of models were created?
Yes, so that is correct. 70% of the models were completed over a 9-year period, and 3 of those years, the Blaschkas were only working on the glass flowers half of their time. Rudolph, after Rudolph's father died, he, as I mentioned, he experimented with his materials and methods, and I think his experimentation, slowed him down a bit just because he was, trying different things. He also, his work was greatly disrupted by World War I. He also, his production was slower, but he created many of, the masterpieces in the collection on his own, and a lot of them are pretty big. He made like many of the largest models were made by Rudolph alone. So I think that kind of accounts for his slower productivity, through the duration of the project.
Mm-hmm. That makes a lot of sense. This is a really good question. Can you create dried herbarium specimens of succulents, or do they need to be preserved differently because of their water content?
Yes, we can create herbarium specimens from succulents. Like, for example, we always plan about a cactus. If we have imagine a globose cactus, we could slice them to fit in in the way we press plants between newspaper and cardboard. This is a way, because the thing is, we have to preserve what is important taxonomically to identify the species. Then sometimes if we cut the plant to fit in the way we preserve them, we still can identify the plant. But there is another way also to preserve cactus, for example. We can dry them in the 3-dimensional shape. Yeah, and then in this way they would not be stored in the cabinets as the other plants. This is not like very good in terms of spatial management. The best way is to try to make all the collections more or less in a standard way. Then we can store them in less space, but it's possible. The thing is, when we are preserving them, we have to take care, like slicing them to fit in the between newspaper, and also it will take more time to preserve, then more time to dry. Then sometimes you, instead of taking like 3 days, as is usually the time that it takes for other plants, we will have to let them dry for 5 or 7 days, and sometimes have to change the newspaper to a new newspaper to help them to dry faster. It's just it's just some details that we will have to pay attention when drying a succulent plant.
Awesome. Thank you. Okay, I'm not seeing any other questions in the chat. If anyone does have any other questions, feel free to email them to me afterwards. You should all have the Arboretum Public Programs email, and we can certainly get those questions out there. Otherwise, thank you all so much. Especially thank you to our panelists for your time today, for your expertise, and also thank you to our wonderful audience members. Hope everyone has a good night.